Hey guys, sorry that this week has been and is going to be so weird. Miss Watson would now normally be in there working with you guys on this stuff, but she unfortunately is not going to be at school this week. And so, as far as today goes, we do need to take a little bit of notes, and they're gonna have to be over video, and I'm really sorry about that. But we are talking about some pretty interesting stuff today, so I think it'll be okay. All right, so we have been talking about how to read DNA, turn it into RNA, take that RNA, make it into a protein, and that end protein is what is used to build and operate most of your body, okay? But today I want to talk about mutations. Now, unfortunately, mutations are not as cool as they are on TV. They're not going to give you like X-ray powers or regenerative bone claws. Most of the time they are just going to give you cancer. And they're also not as glamorous. So a mutation technically is just a change in the base sequence of DNA or mRNA that are coding for a protein. So you can see here, this is what the original sequence is supposed to be, and we have a mutation right here, if that green thing will go away, where that one base that used to be a T or a thymine was mutated and is now a C or a cytosine. Okay, now some of the reasons this might happen are exposure to radiation, exposure to carcinogens, and what is actually going to happen is those chemicals or those energy particles, if it's like the sun, those UV rays are literally going to get at your DNA and they will like knock bases out, and so then it just has to be refilled with whatever is closest. So of course I'm in the wrong spot again. All right, so there are two major kinds of mutations, the first of which is called a point mutation, and a point mutation is just a mutation at one single point. Okay, so a single base is going to mutate, and that is going to then in turn cause the RNA to change, which then is going to affect one single codon. Okay, so I'll show you what these look like here in just a second. But our other type of mutation is called a frame shift. Okay, and it is where the whole sequence, the whole sentence of DNA is changed because the number of bases have changed. So when we're reading DNA, when we're reading RNA, we read it in 3. So we were reading it by those codons. If you insert or delete a base, everything below that is going to shift. That frame that you are reading things by is going to shift. So it's called frame shift. And again, I'll show you what those look like. So I want to first talk about point mutations because there's a couple of different point mutations. So the first, again, a point mutation is just a mutation at one point. So the first point mutation I want to talk about is called a silent point mutation. Okay, so if we look over here, you can see I have TTC in the DNA, which codes for LYS. Okay, now if it got exposed to some sort of mutagen, or I don't know, sniffed paint, or ate too many hot dogs, or somebody blew cigarette smoke in your face, whatever, we can have that DNA mutate and one of those bases can change. Okay, so that would of course change the mRNA. Now in this case, the end protein does not change. So we had a mutation, but it didn't matter. Okay, so those are called silent point mutations, where the mutation causes no change in the amino acid produced. Okay, and this is caused by something called the third base wiggle. Okay, so the third base in a codon, which in this case is T or C, you get a little bit of wiggle room. Okay, so if we look at our chart, let's take serine for example. UCU, UCC, UCA, and UCG all make the same protein. Okay, they all make that same amino acid, and that is due to third base wiggle. That's why sometimes in here you'll have like, UUU and UUC both make phenylalanine, okay? It's because whatever protein this is probably pretty important, so we don't need some simple point mutation screwing with that protein. So we're going to make sure that, you know, it's kind of like a fail-safe. You have backups just in case, okay? So those are silent point mutations where we had a mutation, but nothing really happened. Okay, our next type of point mutation is called a missense point mutation. So if we compare over here to our original DNA, TTC, if we mutated this center base to a C, that's going to change our mRNA, and it is going to change our final protein. Okay, so a missense is where the mutation does change the end amino acid. Now the way that I keep that straight in my head is missense, there has been a mistake, okay? Now, depending on where at in the protein chain this missense mutation is, it can have different effects. If it's at the end, you might just have a little bit of sickle cell anemia. If it's at the beginning, you probably weren't even born. Okay, so it just kind of depends on where at and how important. So sickle cell is a really good example of a missense mutation. This is what a normal red blood cell should look like. They look like fat Cheerios. And it's because they're full of hemoglobin, which is the chemical that absorbs oxygen from your lungs. Okay, now these over here are sickle cell white blood cells, or red blood cells, excuse me. And you can see that they don't look right. Okay, like they're not chunky like these guys over here. It's because of a missense point mutation. And what this does to the person who has this is it means that like just walking around makes them be out of breath because they don't have as many hemoglobins in each white blood cell, so they're not able to absorb oxygen from the air very well. Okay, so moving on. The next type of point mutation is a nonsense point mutation. Okay, so if we have our original DNA here and the first base mutates, sometimes you can insert a stop codon. Okay, so a nonsense point mutation inserts a stop. Now the way that I keep that in my head is nonsense, no proteins. Okay, so we're not making a protein. We're not finishing that protein here. Okay, so again, depending on where this is at in the protein chain, if it is at the beginning, you probably died. If it is at the end, you probably are just like horribly ill, horribly diseased. Kind of depends on what it is and where it's at. But these types of point mutations are pretty devastating because there's not really any part in the protein that is not necessary. So those are point mutations. Okay, one spot was broken. Now I want to move over and talk about frame shifts, okay? So again, we are shifting the frame with which we read these things, okay? So if we look here, I'm reading by threes, I'm reading those codons. Well, if we have removed a base, every codon behind that is different now, okay? So basically, every spot behind the frame shift is ruined, honestly, okay? You're either inserting a base, which causes all of the codons behind to be changed, or you're deleting a base, which causes all of the codons behind to be changed, okay? So I want to show you what an insertion frame shift looks like. So it's where we're adding bases into that sequence. We're inserting a base. So if this is my original sequence right here, and I have inserted this adenine right here between these two cytosines, my first codon will still read like it's supposed to. But you can see that CAC does not code for glycine and CAC does not code for cysteine. So every single protein afterwards is going to be not correct. Now, the really easy way to tell when a frame shift has happened is your number of codons is changed. Your number of bases is changed. We just have this random G out here at the end, and he can't code for anything because there's only one thing. You have to have three to make a code, you have to have three to make a codon, excuse me. So everything after that frame shift is ruined, okay? The opposite, a deletion, is where we have removed a base. So if we take this G and we kick it out, every protein beyond that is affected, okay? Or every amino acid, excuse me, beyond that is affected. And it's really easy to tell because you end up with the wrong amount of codons. So back here we have two bases. That does not make a codon, okay? So those are our different types of mutations. We have silent point we have nonsense point, and we have missense point. And then you have an inserted frame shift and a deleted frame shift. Okay, so those are your 5 types of mutations that you're going to be working with on your assignment for today. Go ahead and make sure that, any notes that you've taken, you can absolutely use those. They're going to help you on the assignment, and just let either me through an email or the sub know if you need anything. Okay, bye guys.